Hello everyone, my name is Neha Reddy. And today I'm going to talk to you about stroke, which is one of the leading causes of disability in the United States. Every year, 800,000 Americans experience a stroke, which is caused when a blocked blood vessel leads to often lasting brain damage. This brain damage can then lead to difficulties with movement and experiencing sensation. And stroke survivors can have challenges doing activities in their daily lives, like brushing teeth and preparing food. For years, researchers and physical therapists have been asking the question, how can we help stroke survivors fully recover their function? But one major challenge is that we don't completely understand how the brain actually changes after a stroke. And the more we can learn about how the brain changes, the better we can develop targeted rehabilitation strategies to address these changes and lead to restoration of function. One important tool that we can use to study brain changes after stroke is called functional MRI, or magnetic resonance imaging. You might have heard of MRI before as a way to take pictures of the way the brain looks, like the one shown here. And functional MRI is a way to take pictures of the way the brain functions. So for example, I can ask somebody to wiggle their fingers and actually take a picture that shows us which parts of the brain are being used while they do that. And the orange regions shown in this image represent the types of areas that could be used from the brain doing something like experiencing sensation. And functional MRI can be a great tool to study the parts of the brain that are being used after a stroke. One key region of the brain to study after stroke is called the brainstem. The brainstem connects the brain to the spinal cord, and it's an important center for the control of movement and sensation. But functional MRI of the brainstem is unfortunately very challenging, and therefore it's pretty uncommon. This is because the brainstem is a fairly small region compared to the rest of the brain, and you can think of it like the pictures we take are sort of blurry there. And for this reason, there's very little functional MRI research of the brainstem after a stroke. Here is where my work comes in. I study the development and testing of new functional MRI methods to make whole brain functional MRI possible, including functional MRI of the brainstem. My work involves developing new techniques that involve new ways to take these MRI pictures and also new methods of analyzing the data. But before I applied these techniques to studying stroke, I first wanted to test how well they'd work in healthy individuals to see how effective they'd be. So I collected functional MRI scans from healthy individuals who experienced sensation of their hands and of their feet. And I wanted to see how well these methods would work to actually identify the small brainstem regions that are involved in sensation. Shown here in this image are the two brainstem regions that we know are involved in sensation. And I compared these regions to the ones that we found in our study, and we found it to be a great match. The location of the regions we found in our study are aligned with where we know the regions are. And for a little more context, the size of each of these regions is about 3 millimeters. That's just the thickness of 2 pennies. So our findings showed that my new techniques worked really well in the brainstem. And in fact, this is the first time that these functional MRI methods have been used to identify these particular brainstem regions. So now I want to go back to my original question, which is, how can we understand more about the way the brain changes after a stroke? My work involves applying these new and promising functional MRI techniques to directly measure whole brain activity while stroke survivors move their arm and their hand, and to see what parts of the brain are actually being used by stroke survivors while they do these movements. My ongoing study involves collecting measures of both muscle activity and brain activity from individuals who've had a stroke. First, I collect measures of muscle activity to see the level of muscle impairment in each individual, and then I relate that muscle activity to what we see in the brain using these new functional MRI techniques. And what we learn from this study can help us to develop new targeted rehabilitation strategies to help restore function, and ultimately to help stroke survivors regain their independence and their freedom to interact fully with the world. Thank you.